Okay, well, we've started. So welcome everyone to What's New in MPMI, which is the new virtual seminar series run by MPMI, where we bring different papers from the journal to you, where you get a chance to actually hear the author present it and ask questions of the author. We're doing this at 2 different times to encourage global participation. This seminar is geared towards people, listeners in around the Pacific, but I know we have people from all over. And our other time in 2 weeks will be at a time that's much more convenient for people around the Atlantic. So let me introduce myself. I'm Jean Harris. I'm the editor-in-chief of MPMI, and I'm very excited to welcome you all here today. We have a very interesting seminar that I've pulled from. This was my editor's pick from our focus issue, which was our January issue, a focus issue examining the cell biology of plant virus and virus vector interactions that was edited, guest edited by Dr. Yi Li, who is joining me here as co-host from Beijing. Yi is a professor at Peking University in Beijing. And in addition to Yi Li, we also had the other guest editors were Tessa Birch-Smith and Aiming Wang, who put together a really lovely focus issue on plant virus cell biology. And both this week's seminar by Hong Lu, who will be talking about her research, and in 2 weeks, our next seminar will be given by Brandon Reagan. He and Tessa Birch-Smith wrote a very interesting review on plasmodesmata, the cell biology of plasmodesmata, and how viruses co-opt that system to move through the plant. That was a review. So this is the first time we will be highlighting a review, but it's been one of the reviews that has attracted the most attention, and I think you will enjoy it all. That will be 2 weeks from now. So we're going to start now. I will turn to Hong Lu, who is coming to us from Chinese Academy of Sciences in Beijing. And Hong Lu will be presenting her research from Feng Shui's lab, examining a very interesting tritrophic interaction between plant, virus, and insect, an aphid, and showing how once you have 3 partners, it gets a lot more complicated, and the virus actually has a symbiotic relationship with its host. So really interesting, a lot of very interesting biology here. So I would like to turn it over to Hong Lu, who will present her research. Thank you.
Thank you. Okay, let me start. Hello, everyone. Hello everyone, I'm Lu Hong. I'm an assistant professor in Institute of Zoology in China. First, I'm very glad so many people are very interested in my article. Cellular gland is the key organ to recognize and accept host plants. Insect saliva proteins can modulate plant defenses and facilitate feeding. My major research is exploring the function of cellular gland or saliva proteins in insect-plant interaction and plant virus transmission. Plants, viruses, and insects function in a complex tripartite interaction. In this topic, I present an interesting twist on this interaction. An insect symbiotic virus can facilitate aphid adaptation for aphid adaptation to host plants by suppressing jasmonic acid responses. Aphids are agricultural pests. They suck seed elements of host plants and transmit plant viruses. They can cause great economic losses of crop production. Aphids possess symbiotic viruses, but only several symbiotic viruses have been reported and characterized, such as these 6 RNA viruses: APV, HPV, BRBV, RAV, APGLV, and 2, and MEV1. And these 2 DNA viruses, MPDNV and DPLDNV. However, the effects of symbiotic viruses on aphids and the mechanism involved were largely unknown. Plant-mediated horizontal transmission is a prevailing characteristic of aphid symbiotic viruses such as IHPV, MPDNV, DPLDNV, RAV, and MEV1. In this figure, when virus-free aphids were transferred to plant levels fed by high-virus aphids, and then the virus-free aphids become virus-positive. Just because these viruses can be transferred to plants via injection with aphid saliva. When aphids feeding host plants, they can inject saliva proteins into plants. These saliva proteins can exert certain influences on plants, such as regulating the aphid-plant interaction. With hypothesis, the symbiotic viruses can function as aphid effectors, can modulate the aphid-plant interaction. In our previous work, the fitness of PFS was highest on Visia faba, followed by Medicago tractella and Visia volosa, and finally Medicago sativa. And on Medicago sativa, the PFS didn't survive. In previous transcriptome, salivary transcriptomic data, we found the number of host-specific differentially expressed salivary gland genes was negatively correlated with the fitness of aphids on this host plant. In our transcriptomic sequence of salivary glands, we found as up to 13% of these can be mapped to the genome of APV. APV was a picorna-like positive sense single standard RNA virus. Its genome has been successfully sequenced. It has 2 large open reading frames encoding helices, protease, RDRP, and CP. We put forward a scientific question: what's the function of APV involved in the aphid-plant interaction? In our results, APV particles were extracted from PFS and observed by electronic microscopy. And APV was primarily located in cellular glands and gas, but it is almost undetected in oris. And APV had propagated in PAVs. The vertical transmission rate of APV was very low. Only 33% of the progeny was APV positive. Just because the high abundance of APV in cellular glands, the horizontal transmission rate of APV was also investigated. APV is horizontally transmitted to host plants during aphid feeding, but the virus didn't replicate in plants. The host race of PAVs exhibits the highest fitness on Visia faba, followed by Medicago tractula and Visia volosa. When PAVs were adapted to low fitness host plants, more APV was produced in aphids and horizontal transmitted to plants. To explore the effects of APV on aphid adaptation to low fitness host plants, the survival and the reproduction of APV infected and APV free aphids was compared. On low fitness host plants, APV was beneficial for the survival of PAVs. This benefit of APV would likely be derived from either a direct effect on aphids or an indirect effect on aphids via the plants. From the direct effect on aphids, the molecular response of aphids to APV infection was investigated by comparing the transcriptomes of APV infected and APV free aphids living on Visia volosa. Among these 21,725 genes, only 184 genes exhibited differential expression. 14 genes were upregulated and 117 genes were downregulated in APV-infected aphids. The highest fold change was only 4-fold. Moreover, the APG technique was used to characterize the aphid feeding behavior to APV infection. APV-infected aphids spend less time on NP and C waveform and spend more time in F waveform. No significant variation in the time spent in E1 and E2 and G waveforms. Combined with the molecular response and the feeding behavior of aphids, APV had a marginal effect on aphids. Plant hormone in signaling plays a key role in aphid-plant interaction. Jasmonic acid confers the defense against insects. Salicylic confers the resistance to biotrophic pathogens. On low fitness host plant Visia volosa, a a significant decrease in the levels of GE and GAI solution was observed from after APV infected aphids feeding. The amount of assay was also increased. It indicated that APV can lower the level of GE to facilitate aphids level on low-fitness host plants. We summarized APV can be considered to be aphid effectors. It can facilitate host aphid adaptation to unsuitable plants by modifying the fetal hormone defense response. Our research demonstrates a novel case of symbiotic virus-mediated three-species interaction. In the end, I would like to express my sincere thanks to my advisor, Kanglo, and advisor, Dr. Cui Feng, and our team members. And thank you very much for your listening. Thank you.
Thank you very much, Hong, for a very interesting presentation. We have some questions already coming in the Q&A box, and if anybody is interested, you can write your questions down there, and Dr. Yi Li and I will read them. Sorry, I'm getting it so I can see everybody again. We have our first question by Andres Ricardo Peraza Arias, who says, please tell me more about the methodology. So, for example, the number of aphids for a sample, and is it possible that the response is different according to growth and development of the plant? So it's really two questions. So maybe the first question, Hong, is the number of aphids that are on each plant. The number of insects.
你用了多少个蚜虫吗？
这是存活的那部分。
你可以问一下。You can ask number of individuals or sample. 就是你一个样品去一个样品用了多少个蚜虫
？Is this the survival rate and all reproduction?
I think the question is how many insects, how many aphids?
For one replicate.
Yes.
一个重复多少个蚜虫
？13 to 14.
30 to 40.
30 to 40.
40
per plant. Okay. And then the second half of that question is it possible that the response is different depending on the development of the plant?
因为这个植物，对吧？根据这个植物的发育，还有生长的不同的蚜虫的反应，对这个不同的植物反应相同，有什么区别吗？
老洪，你听懂这个问题了吗？他说一就是这个植物的生长的状态或者发育的状态是不是对蚜虫有不同的影响
？There is no difference different response according to growth and development of plant.
Okay. Yi, do you want to read the next question?
Yeah, next question is from Karen Alvia. Two questions. Is the life history of traits of the aphid infected with EPV same as that of non-infected in host plants？ 就是这个蚜虫，有病毒的蚜虫跟没有病毒的蚜虫，它们的那个就是行为啊、存活啊有什么变化没有？
On low-fitness host plant Visia volusa, the APV has a beneficial effect on the survival of aphids. APV survival much better. APV infected aphids survival much better than APV free aphids.
Okay,
but on best fitness,
high fitness, yeah.
But on the good fitness host plant Visia faba, the APV infected, the survival of APV infected and APV free aphids was no difference, has no difference.
So no difference on life history, so no difference on timing of aphid development? Like, does it affect how fast they develop?
哎呀，那个罗红，那个今天的问题是，就是在那个那个适应性非常好那个植物上，对蚜虫的发育啊、生长的有没有什么影响？就除了死亡率以外。
No difference.
No difference.
对 no difference.
Okay. So Karen Alviar has a second question. She asks, were there validation experiments after transcriptomics? Did you do QRT-PCR on any of the genes?
In the transcriptomic data, we found I have no experiments on this.
你定量验证那个转录组的结果了吗？
No.
So you did not do a qPCR to confirm the sequencing results. For example, to pick up some specific genes, then do a qPCR. Have you did that?
没做一些基因，选一些基因去做 qPCR 验证吗？
没有。
做过吧？做过。你选一些基因做验证吗？或者是你那个做的那些基因，然后去验证了它
？None of them shows some gene for validation.
I have some different. Some many different genes was selected to for this experiments, but we 没放进去。
刘红，这就告诉我，就是说有没有选一些特定的基因做一个 qPCR 验证你那个测序的结果？
你是主要做了一些你的其他的基因吧？就是你那个和通路相关那些基因都做了吧？
英中文，
然后李老师给翻译一下。
李老师，我主要是做一些那个，就是关于我那个植物防御方面的那些基因定量验证。
哦，
就是莫蒂沙西和通路的。
做了，崔老师说做了，你没放文章里。
对，我做了一些定量验证，但是我没有放到文章里面。
我问你是做这个 GA 信号通路的是吧？
对 GA 跟 SA 的信号通路的定量验证，做了没有放到文章里面，就是直接用激素去代替激素。
So Hong has picked up some of GAE or some SE pathway genes made the qPCR, but she did not put this result into the paper, but instead she used the quantitative amount of the GAE or SE to measure the GAE SE level.
Okay, thank you. So Good Luck Benjamin is asking, is there data on metabolites activities during the course of the interaction? Thinking about first, primary and secondary metabolites.
听懂吗，罗红？他问你这个次生代谢产物有什么变化没有？对。
你说。
次生代谢产物。
有做这方面吗？
有做。
没只做了 GA 的，没有做其他次生代谢的。
我们只做了 GA 跟 SA 的，没有做其他的代谢活性的。
用英文说，用李老师来回答。
Okay. The answer is she only measures GAE and SE, but she did not measure other metabolisms.
That could be a very interesting direction for the future, looking at the other metabolites. I agree, it's a very interesting question, especially considering all the hormone changes. Okay, yeah, I'll let you have the next one. I think.
Okay, where is this? I cannot find it.
Oh,
in the
Q&A. Did the box go away? You can click Q&A. It should be, I think, I don't know how to pronounce. Where Tsui? I can read that one. Where Tsui? Yeah.
感染增加了蚜虫的存活率，但是减少了它的生殖率，对吗？
So where Tsui asks, APV infection increased the survival rate of aphids, but it decreased their reproduction rate, correct? And is the difference significant or not? And if so, do you have any idea why?
你有一些那个想法吗？为什么它增加了它的生殖率，然后减少了它的这个题呢？
题呢？
往上滑一下 ，slide。
Oh, I think that one went into answered.
对，邱老师对生殖有影响。
看见题了。
你就是说你对这个
不用你直接回答。
Yes, APV infection decreased their reproduction rate.
Do you have an idea why that maybe happened? Do you have an answer?
你有什么想法吗？为什么会发生这种事？
有什么想法？
生殖，
为什么增加了这个生殖？
怎么说？你帮我翻译一下。
就是
，我想说的是，就是 APV 感染之后要保证 aphid 的，然后降低它的就繁殖率，是保证它体内的能。
So this may be a trade-off that between the reproduction and the APV infection.
Yes.
Interesting. I wonder what the mechanism is.
机制，这个有可能的机制是什么？
机制是什么？
Do you have any idea about the mechanism of this trade-off?
机制。
喂，
You, I don't know.
This can still be unknown. That's okay.
Yeah, still under question.
Yeah. Why don't we move on to the next question? Yi, do you want to ask? We have one from Steve Merrick.
Jenny, I could not find where the question in my screen.
Oh, okay. No problem.
Yeah.
You're helping a lot. Don't worry. Stephen, it's right across the bottom. There's a Q&A. You can click that. That's okay. I can read them. Stephen Merrick asks, do APV-infected aphids thus have a broader plant host range than non-APV-infected aphids? It's an interesting question. If they're more successful on plants that are not as good a host, does that mean they can spread more broadly?
Yes, I think APV infected have a broad plant host range than non-APV infected aphids. Because APV infected aphids can help APV survival on low fitness host plants. Can increase the plant host range.
What do you mean?
Interesting.
它会提高它的在其他植物上的存活，就是可以在其他植物上活。
还有更广泛的技术范围吗？
I guess.
Yes.
I guess it has.
You guess or you sure? It has a more broader plant host range. 就是 APV 感染的是不是有更广泛的寄主范围
？APV 感染的
Yes or no?
技术的
Yes. Yes.
So we're working through all the questions. Yi, do you have a question for Hong?
Yeah. So Hong, the question is APV infected aphids is increase the aphids fitness on host, or it have more branched broad host. 这是两个问题，就是说带病毒的蚜虫，可以取食更多的植物，或者在更多的植物上生存很好，还是带病毒的蚜虫在这个提高的它在植物上的生存的适应度，可以这样讲，是吧
？Yes.
是，我觉得它这个寄主范围可能没变化。不过是提高了在基础上适应的那个程度，因为它降低了墨鱼酸。
对，它是是它提高了它的存活能力，应该是。
Okay, so the answer for this question is, APV infected aphids have better fitness in the plants, but did not increase the host range.
Oh, yeah. Okay. Interesting. So that's an interesting point. So they still are infecting the same kinds of plants. They just are have better fitness there.
And yeah.
And so how are you measuring fitness? Because fitness is sometimes measured by number of offspring, right? Number of progeny. And in this case, you said reproduction is decreased. So what is your measure of fitness?
你对于适合度， 适合度是用什么方法测量的？然后是用树木呀，还是后代的？
低适合度和高适合度。之前就是你scientific report 的那个文章，你发现比较这几种植物，它的你有几个指标，比如说种群的那个增长率啊、后代啊什么的。
后代的树木。
In my previous paper, we found 3.1 life 就是生命 life table and the feeding behavior and the transcriptomic of cellular glands. From this point, we found the fitness of PFS on these three host plants. The fitness host plants exhibited the highest on VCF faba, 然后 the lowest followed by Medicago tractula and VCF ulusa.
你先想生长的构成，就是种群增长率什么的。
The life history includes 5 points, including the net production rate and 净生殖率、总存增长率。
郭伟帮他翻译一下。
You have 5 parameters to measure in the life table. So the first parameter is
net production rate.
the second parameter.
The second is
你说汉语。
就是平均世代周期。
Average life cycle.
The mean generation period.
Life cycle.
The
Generation time or generation length?
Range. The generation range.
中文说
中文，卢红。
有平均世代周期，然后净生殖率以及
郭伟翻译一下。
世代周期。
然后
The average duration of the life cycle. 还有个什么？
How long they live.
第三个是什么？ The third parameter is 中文是啥？
就是
差不多就行了，不一定非得补个，差不多说了这个。
生存时间。
The duration of the life. Okay. 寿命。
对 ，life。
lifespan.
Yeah, lifespan.
然后and production 就是繁殖率 production rate。
production rate.
就是 life table includes this 5.
How many progenies can you produce?
Right. That's what I'm thinking.
Yes.
Okay. So is that increased in the APV aphids?
是在 APV 感染的那个
increased progeny?
你可以直接说他们都
是在那个 APV 感染的那个
后代有 APV 后代增加了，数量增加了。
减少了 decrease.
A decrease.
Decrease.
So you're measuring fitness not by number of progeny, but by lifespan and by like feeding parameters. Yeah. Let's see. Have we missed any more questions? I think we have.
Jay, maybe I have a question for Lo Hong.
Yes, please do.
Yeah. So, 然后又说是发现 that the APV infect aphids decrease the GA in the plants.
Yeah.
Yeah, it's the but without APV virus, aphids do not decrease the GA level. Yeah, so that means the APV somehow did this job. Yeah. So do you know how APV manipulates the GA level in the plant?
就他知道
APV 怎么降低了 GA 的那个 level 水平？
是我怎么知道吗？
对啊，就是你猜测。
你怎么想？
怎么想的？
不知道。
The mechanism of this was unknown.
And okay, so from you did the transcriptome assay of the APV infect aphids on plants and the no APV in plants. Have you compared these results? Found something changed, some genes upregulated, some maybe downregulated. Did you find some interesting genes maybe involved in this process?
感染和没感染的 APV 的这个转录组相互比较，有没有和茉莉酸 A 相关的通路的基因发现，然后发生了变化？
这不是昆虫的吗？
不，这是昆虫的。我跟你说错了，是昆虫基因有意思的变化的基因，你发现过吗？你可以说一下，我们曾经做了抗菌肽是吧？但是那个感染的效果不太好。
We found in this transcriptomic data, we found the 抗菌肽怎么说
？Antibacterial
peptide, but it's no difference.
干扰率很低，只有40%左右。
Moreover, the interference rate of APV is very, very low.
不是的干扰率，是那个
The interference rate of aphid genes was very, very low. We found
30% around. 有30%左右。
Only 13%. We found
the efficiency of the RNAi of the antibacterial peptide is only about 30%. So no obvious phenotype were observed after this RNAi.
Okay, yeah, maybe this is usual question because we know that in rice, virus infection could stimulate the J level, J pathway. So in your case, J is down-regulated. So maybe in the future, you can try to find out how virus manipulate the J and the acid because salicylic acid level is increased without viral infection. Yeah. Okay, that's good. Yeah. Okay.
Yeah. We have we have one more question from Michael Jones, who has, it's very related, asks, is there any identity between the APV genome sequence and JA pathway genes? Could there be an RNAi mediated pathway reducing JA expression? So is there part of the APV genome that interferes with expression of JA genes via RNAi? So it's an interesting question. Have you noticed anything?
比较过 APV 的基因组与 GA 的基因有没有相似性？我们从来没有比较过，这是个非常好的 idea。
This is a good idea, but I have no experiments on this adjective.
Yeah, be interesting. Yeah.
Didn't compare the APV genome to the GA pathway genes. So this is a very good idea for your future research.
Yeah, I think it's a very good idea. Interesting.
Yeah.
Good. Well, I think we have one more comment in the chat as well. Okay. Well, wonderful. I think this has been a great set of questions. And thank you also, Yi, for helping and also for, I'm sorry, Feng Shui, Feng Zhang. Oh, I made that. Feng Zhang. Thank you also for helping. And Yi for helping with the questions. So unless anybody has another question, I think we'll stop there. Do we have any last questions? I'm not seeing any. Okay, well, I see some thank yous coming up in the chat. So thank you very much, Hong Lu, for the interesting talk. I'm seeing many thank yous, Patricia Santos, Karen Alviar, Jayani Kalinga, lots of people are saying thank you. So thank you for all joining us here for What's New in MPMI. And next week, oh, wait, we just have, oops, I think I have a question here from, I don't want to leave too soon. There's one question from Brandon Reagan.
2 weeks.
Oh, yes. Good catch, Brandon. That's because this was rescheduled. Thank you. Let me get back to Zoom here. So yes, I just want to correct a mistake I made, which is that our next talk is not 2 weeks from now. It was 2 weeks from last week.
1 week.
So it's in 1 week. In one week,
yeah.
we have Brandon Reagan's talk. Thank you, Brandon. I'm still back where we were last week with when we had the first seminar. So yes, Brandon Reagan and the work from Brandon Reagan and Tessa Birch-Smith, a very lovely review article on plasmodesmata and viruses, the cell biology of it, is going to be next week's talk. Next week's talk, we're going to shift the time. It's going to go back to our 11 a.m. time, which will be less convenient for Asia. Sorry about that. But we will have this all recorded. And that also reminds me that we have all of these recorded. All of these are free. And if you go to the same place where you registered for this seminar, you can get the links to the recordings. Would you have a chance to paste that into the chat, Ashley? Or maybe it's already in there. But if you could paste in where people go to get the recording, that would be great. And that's also where you can sign up for all future seminars. So I hope that you'll join us in 1 week. Recordings are free. And of course, we have opened up Hong Lu's paper, open access. And we have also done the same for Brandon Reagan's talk for next week's talk. Thanks, Brandon, for that catch. And if you look in the chat, you'll see that Ashley has posted the link for how to get the recordings as well as the list of seminars. So I look forward to having you next week. And thank you very much for your attendance.